Welcome back, everybody, for another deep dive. This time, we're going to be taking a look at a paper from 1951
Oh, well.
called Protein Measurement with the Folin Phenol Reagent.
Okay.
And, as kind of dry as that sounds,
Yeah.
this is a really, really cool paper, and it had some really interesting ramifications for biochemistry.
Yeah, it's a landmark paper for sure.
Yeah, and I mean, just to kind of set the stage,
Right.
before this came out, measuring protein was a real problem.
It was a huge problem.
And
Right.
and so this really kind of revolutionized how we were able to study proteins.
Yeah, it's sort of like if you're trying to study something, but you can't really uh, see it very well or measure it very well,
Right.
you're not really going to get very far.
Yeah, you can't really study it.
And so this method really kind of, um, opened up this whole new world of being able to look at proteins and quantify them.
Yeah, I mean, can you imagine trying to do any kind of research on
Yeah.
something that you can't even
Yes.
see or quantify?
Very bad.
It's impossible.
It's like trying to study the wind, without being able to see
Right.
how fast it's moving or what direction.
So how did this method solve this problem? I mean, what was so special about it?
Well, it was a combination of things.
Mm.
Really, first of all, it was surprisingly simple.
Okay.
Like the reagents and the steps involved were fairly straightforward, even for the time.
Okay.
But it was also incredibly sensitive.
Oh, wow.
So you could use it to measure very tiny amounts of protein,
Interesting.
which was a huge advantage over previous methods.
So it's like they figured out a way to kind of amplify the signal of the protein, so to speak.
Exactly. Yeah, it's like they found a way to make the protein shout a little louder.
I like that.
Hmm.
Okay, so let's break down how this method actually works.
Sure.
The paper describes this two-step process,
Yeah.
and the first step involves something I think is really cool.
Okay.
Copper.
Oh, yeah, copper.
So you take your protein, you mix it with copper in a solution that's alkaline,
Right.
which basically just means it has a higher pH.
Okay.
And then something really interesting happens.
Yeah, what happens is the color of the solution intensifies dramatically.
Oh, wow.
It's almost as if the copper is acting like a spotlight,
Oh, okay.
making even tiny amounts of protein visible.
So it makes it easier to see?
Much easier to see.
Yeah. And
mean-
measure.
Got it. And so this is all happening in the first step?
Right, this is all in the first step.
Okay.
So you've got your protein,
Right.
you've added the copper,
Yeah.
you've made the solution alkaline, and boom, you get this big color change.
Oh, that's cool.
Mhm.
And, I mean, just out of curiosity, was this a known thing before this research?
Well, scientists knew that copper could react with certain molecules to produce color changes.
Okay.
But what was really groundbreaking here was the way they harnessed this reaction specifically for protein measurement.
Gotcha. So they kind of optimized it.
Yeah, they figured out the exact conditions and the specific type of copper compound that would give the most sensitive and reliable results for proteins. So they weren't just throwing copper at the problem. They were very precise about it.
Exactly. They were very systematic and meticulous in their approach,
Yeah.
which is what makes this research so impressive.
But it gets even more interesting. The researchers actually figured out that the copper wasn't just hanging around.
Yeah.
It was binding to the protein,
Right.
forming this copper protein complex.
That's right.
And it's this binding that somehow amplifies the color even more.
It's like the copper is grabbing onto the protein and saying, hey, look at me.
That's a great way to put it. So it's not just that the copper makes the protein easier to see,
Right.
it's that it's interacting with the protein in this very specific way.
Exactly. And that's a key point.
And they didn't stop there. They even went on to calculate what's called a dissociation constant,
Wow.
which essentially tells you how strongly the copper is holding onto the protein.
Right. It's a measure of the binding strength.
Okay, so this was pretty high-level analysis for the time.
Oh, absolutely. This level of detail was really groundbreaking back then.
And what did this dissociation constant tell them about the copper-protein interaction?
It showed that the binding was pretty strong,
Okay.
which is important because it means that the copper isn't just randomly attaching and detaching from the protein.
Right, it's got a good grip.
Exactly. It's sticking around long enough to really amplify that color signal.
That makes sense. And this whole idea that the copper is binding to the protein, it makes me think that maybe not all parts of the protein are equally involved in this binding.
You're on the right track there.
Hmm.
They actually found that only about half of the potential copper binding sites on a protein
Wow.
actually contribute to this color boosting effect.
So it's like the copper has a favorite spot to latch onto.
That's a great way to think about it.
So we've got our protein all dressed up with copper,
Right.
glowing brightly. What happens next in this two-step process?
Well, that's when the star of the show enters the Folin Phenol reagent.
Okay, I've heard of this.
Yeah.
I've heard of this Folin reagent. It's kind of a big deal in biochemistry, right?
Oh, it's a classic reagent for sure. It's been used in countless experiments over the years.
Okay, so how does it fit into this protein measurement method?
So when you mix this reagent with the copper-treated protein,
Okay.
a reduction reaction happens.
Okay.
Essentially, electrons are transferred between molecules,
Okay.
and this leads to even more intense color development.
Wow, so it's like a double whammy of color amplification.
Except first, the copper boosts the color,
Uh-huh.
and then the Folin reagent comes in and cranks it up even further.
I like it.
Yeah.
But I'm sensing there's a catch. Something about timing being crucial.
You're absolutely right. Timing is key here.
Okay.
For this whole reaction to work its magic, the pH has to be just right around 10,
Okay.
and the Folin reagent is only reactive for a short window of time.
Oh, really?
Yeah, the researchers actually emphasized the need for quick and thorough mixing in the paper.
So I can just imagine them like frantically swirling these test tubes,
Yeah.
racing against the clock.
It was probably pretty intense in the lab.
And I have to ask, what happens after the Folin reagent does its thing? Does the color just stay put?
That's actually a great question, and the answer is no.
Oh.
The color continues to deepen even after the reagent itself becomes inactive.
What?
Yeah, it's kind of like a chemical afterglow.
Really?
They theorized that this might be due to some rearrangement of the molecules formed during that initial reduction reaction.
So even though the reagent is kind of spent,
Right.
the color is still developing.
Yeah, it's like the reaction has this kind of momentum that keeps it going for a little while.
So this whole process, it's kind of like a carefully choreographed dance,
It really is. Yeah.
but with molecules instead of dancers.
Exactly. And the timing and the steps all have to be just right to get the best results.
And you mentioned earlier that copper binds to proteins in a very specific way.
Yes.
Did the researchers find that this affected how different proteins reacted to this whole color-changing process?
That's a great question. And it leads us to one of the most fascinating aspects of this research. The idea of what we might call protein personalities.
Protein personalities. I like where this is going.
Yeah, it's a pretty cool concept.
Okay. I'm hooked. Tell me more.
We'll have to save that for after the break.
No, come on.
Don't worry, it'll be worth the wait.
All right, fine. But you better deliver.
I promise I will.
We'll be right back after a quick message.
All right, so protein personalities.
Yes, you left us hanging with that one.
Yeah, sorry about that.
Okay, so what do you mean by protein personality?
Well, what the researchers found was that different proteins actually produce different intensities of color
Really?
with this method.
Yeah.
Yeah.
So it's not a one-size-fits-all kind of reaction.
Exactly. It's not like every protein gives you the same shade of blue or whatever.
So it's like each protein has its own unique way of interacting with the Folin reagent and the copper,
Right.
almost like a fingerprint.
Yeah, you could think of it that way. Like some proteins are really good at binding to the copper
Mm-hmm.
and others not so much.
And this affects how much color they produce.
Exactly. And they found some pretty striking differences too.
Oh, like
Well, for example, they found that trypsin,
trypsin.
yeah, it's an enzyme.
Okay.
It creates a very strong, vivid color reaction,
Oh, wow.
whereas gelatin produces a much weaker response.
Interesting. So it's like some proteins are really enthusiastic about this color-changing party,
Yeah,
and others are kind of wallflowers.
exactly. That's a great way to put it.
So this discovery of protein personalities, did it have any practical implications for using this method?
It definitely did.
Oh, cool.
One important finding was that this difference in color intensity is less pronounced when you're dealing with complex mixtures of proteins,
Oh, okay.
like those you'd find in tissues.
So like in a real-world biological sample.
Right, exactly.
Hmm.
So while individual proteins might have their own quirks in a mixed group, they tend to behave more similarly.
That makes sense. It's like the individual personalities kind of blend together a bit in a crowd.
Yeah, you could say that.
But even with these variations in color intensity, the method was still incredibly powerful, right?
Absolutely.
Especially for analyzing really tiny amounts of protein.
Yeah, in fact, the researchers took this method to the next level by developing a microanalysis technique.
A microanalysis technique, what's that?
So basically, they figured out a way to do this whole protein measurement thing, but using incredibly small volumes.
Like how small are we talking here?
I think microdrops barely visible to the naked eye.
Wow. So they weren't just content with revolutionizing protein measurement on a larger scale.
Nope.
They wanted to push the limits of what was possible.
Exactly. They were real pioneers in the field of microanalysis.
So how much protein could they actually measure with this microanalysis technique?
Well, they were able to accurately measure as little as 0.2 micrograms of protein.
0.2 micrograms. And that's considered accurate.
Yeah, for the time, it was incredibly accurate. To put
Yeah.
that into perspective, that's like trying to weigh a single grain of sand.
That's mind-blowing. And to do
Yeah.
that with such precision back in 1951,
I know, it's really impressive.
so they're working with these minuscule samples, pushing the boundaries of sensitivity, and what happens?
Well, of course, there were some challenges along the way.
Oh, I bet.
One of the biggest surprises was that in these tiny volumes, the amount of color produced was actually less than what you'd expect based on the larger scale reaction.
Oh, that's interesting. It's like the rules of the game were changing at the micro level.
Yeah, it was a real head scratcher for them at first.
So what did they do? Did they just give up?
Oh no, they were not quitters.
Hmm.
These researchers were determined to get to the bottom of it.
I like that.
And they eventually discovered that using slender tubes instead of wider ones,
Slender pubes?
yeah, like really narrow test tubes,
Okay.
helped to solve the mystery.
So how did that make a difference?
Well, it turns out that by controlling the geometry of the reaction vessel, they were able to minimize certain surface effects.
Surface effects.
Yeah, basically the shape of the container was affecting how the reaction was happening.
Oh, wow. I never would have thought of that.
Yeah, it's one of those things that you don't really appreciate until you start working at these really small scales.
So by using these slender tubes, they were able to get accurate results even with these tiny volumes.
Exactly. They had to kind of tweak the setup, but they got it to work.
That's so cool. So it's like they had to adjust the choreography of the dance floor to accommodate the smaller group of dancers.
That's one way to put it.
But let's step back for a moment
Okay.
and think about the bigger picture.
Right.
This research was published way back in 1951. Why should we still care about it today?
That's a great question.
Yeah.
And I think the answer is that the impact of this research really cannot be overstated.
Okay.
It provided scientists with a reliable, sensitive, and relatively simple tool to quantify protein,
Okay.
which is essential to countless areas of research.
Like what?
Oh, from basic biology and medicine to agriculture and even food science.
It's like they gave researchers a new set of eyes to see the invisible world of proteins.
Exactly. And it opened up countless possibilities for exploration and discovery.
And to think this all started with a paper from 1951.
It's amazing, isn't it?
So you're telling me this method is still used today?
Oh, absolutely.
Yeah.
It's still a standard technique in many labs around the world.
Wow, so it's a true classic.
I'd say so. And it reminds us that even research that might seem old can still hold incredibly valuable insights
That's a good point.
and can serve as inspiration for new discoveries.
Absolutely. But I'm still really curious about this idea of protein personalities.
Yeah.
The researchers found that different proteins have these unique color signatures when reacting with the Folin reagent. What does that tell us about proteins themselves?
That's a really intriguing question. And to be honest, it's one that researchers are still exploring today.
Really? So this wasn't just a cool quirk, it opened up new avenues of research.
Exactly. It sparked a whole new wave of inquiry into the individual characteristics of proteins and how those characteristics might influence their roles in biological processes.
So it's like realizing that each player on a team has their own unique skills and strengths,
Right.
and that understanding those individual talents is crucial for the team's success.
I like that analogy.
So this focus on individual protein characteristics, it must have had some pretty big implications, right?
Oh, definitely.
Yeah.
It had ripple effects across various fields.
Like what?
Well, for instance, in enzymology, scientists began to explore how these unique characteristics might explain why certain enzymes were more efficient than others, or why they preferred specific substrates.
It's like going from using a sledgehammer to using a scalpel.
Exactly. And this shift towards more targeted therapies has led to the development of many life-saving drugs.
Wow. So we started with a seemingly simple method for measuring protein,
Uh-huh.
and now we're talking about personalized medicine and groundbreaking drug
It
discoveries.
just goes to show you how fundamental research, even something published back in 1951,
Right.
can have such far-reaching and unexpected impacts.
It definitely makes you wonder what other hidden secrets are waiting to be uncovered in the vast world of scientific literature.
That's the beauty of science. It's a never-ending journey of discovery. And this research, with its elegant method and profound implications, serves as a reminder that each new finding has the potential to unlock countless more.
Well said. So to wrap up this deep dive, if you
Yeah.
had one key takeaway for our listener, what would it be?
I'd say it's this. Never underestimate the power of a simple question or the potential impact of a seemingly basic scientific finding. Curiosity and meticulous research can lead to breakthroughs that shape the future.
Couldn't have put it better myself.
Yeah.
And to our listener, if you're feeling inspired to explore further, I encourage you to delve into the original research paper by Lowry and his colleagues. It's a fascinating read, even for those without a scientific background. You can find a link to it in the show notes. Thanks for joining us on this deep dive into the world of protein personalities. Until next time, keep exploring. So this wasn't just a cool quirk. It actually, led to some pretty big changes in how we think about proteins.
Oh, yeah, for sure. I mean, before this, it was kind of like proteins were just these generic blobs of molecules. They all kind of look the same. But this research showed that they had individual characteristics that could be measured and studied, and that was huge.
So each protein has its own personality, so to speak.
Exactly.
Yeah.
And more importantly, those personalities actually mattered.
Okay, how so?
Well, researchers realized that these subtle differences in their chemical makeup could potentially have a big impact on how they function.
But it's not just about how much protein is there.
Right.
It's also about what kind of protein it is.
Exactly. It's like, with people, we all have different personalities, and those personalities influence how we interact with the world.
Right.
Proteins are kind of the same way.
So this realization must have had some pretty big ripple effects across different fields of research.
Oh, absolutely. It really changed the game in a lot of ways.
Like how?
Well, for instance, in enzymology.
Enzymology. That's the study of enzymes, right?
Exactly. So enzymes are these proteins that speed up chemical reactions. And scientists started to explore how these unique characteristics of different enzymes might explain why some enzymes were more efficient than others. Or why they preferred to work on specific molecules.
So it's like each enzyme has its own preferred way of doing things?
Yeah. Kind of like how some people are really good at math and others are more artistic.
Yeah.
And this knowledge about enzymes, I'm guessing it had some practical applications too.
Oh, yeah, definitely. I mean, one of the biggest areas where this research had an impact was in the development of new drugs.
Drugs.
Oh, wow. How so?
Well, before this, it was kind of like, if you wanted to block the activity of a protein, you just had to kind of throw a bunch of stuff at it and hope something stuck.
Right. It was a very blunt approach.
Exactly.
Yeah.
But once scientists started to understand that proteins had these unique features, they could start to design drugs that specifically targeted those features.
So it's like instead of trying to shut down the whole protein, you could just target the part of it that was causing the problem.
Exactly. It's like going from using a sledgehammer to using a scalpel.
That's a great analogy. So this must have led to some more effective drugs.
Oh, absolutely.
Yeah.
This shift towards more targeted therapies has led to the development of many life-saving drugs, particularly in areas like cancer and autoimmune diseases.
That's incredible. So we started with a seemingly simple method for measuring protein, and now we're talking about personalized medicine and groundbreaking drug discoveries.
It's amazing, isn't it, how something that seems so basic can have such far-reaching consequences.
It really highlights how important fundamental research is.
Absolutely. Even research that's, over 70 years old can still be incredibly relevant today.
It definitely makes you wonder what other hidden gems are out there.
Yeah, who knows what amazing discoveries are waiting to be made.
Well, that's a great note to end on. So to wrap things up for today, we've been talking about a groundbreaking paper from 1951 that revolutionized the way we measure and understand proteins.
A true classic.
We've seen how this research led to the development of more sensitive and precise analytical techniques, and how it sparked a whole new way of thinking about proteins as individual entities with unique characteristics.
Protein personalities.
Exactly. And we've seen how this knowledge has had a profound impact on fields like enzymology and drug development.
It really has been a game changer.
So to our listeners out there, if you're interested in learning more about this fascinating piece of scientific history, we encourage you to check out the original paper by Lowry and his colleagues.
You won't regret it.
It's a surprisingly engaging read, even if you don't have a background in biochemistry.
Absolutely.
And who knows, maybe it'll inspire you to make some groundbreaking discoveries of your own.
That would be awesome.
Thanks for joining us on this deep dive into the world of protein personalities.
It was a total pleasure.
Until next time, keep exploring.